Hello, everyone. Welcome back to the GEP Pathways video series. Today, we will be working on part 6.3. This diagram here shows us an overview of the splicing process. During this, introns are removed and the adjacent exons join together, creating our exon junctions. In general, when looking at our splice sites, we give them a phase, which can be 0, 1, or 2. For our donor site, this phase is determined by the number of nucleotides between our last complete codon and our GT donor. As for our acceptor site, the phase is determined by the number of nucleotides between our first complete codon and our AG acceptor. When we are identifying the phase of our splice sites, we need to be sure we're looking in the proper reading frame that corresponds to the coding exon that we are looking at. In addition to determining our splice site phases, we must also make sure that they are compatible with each other. The splice donor site of one CDS will pair with the splice acceptor site of the next CDS. The pairing of our splice site phases must always equal 0 or 3, meaning if we have a donor phase of 0, our acceptor phase should be 0. If we have a donor phase of 1, our acceptor phase should be 2, and if we have a donor phase of 2, our acceptor phase should be 1. This is because when our introns are spliced out, the joining of our exons must create either a full codon, which would be 3 nucleotides, or no codon at all, which would be 0 nucleotides. For a more in-depth video about splicing, see this video here. Because the T-BLAST-N alignment for CDS1 of Reb terminates at 19,150,856, we expect to find the splice donor site for CDS1 at around that position. To examine the genomic regions surrounding the splice donor site of CDS1, enter NC underscore 052530 colon 19,150,856 into the chromosome range or search terms text box. Then click on the Go button and zoom out 3 times and another 10 times to examine the 30 base pairs surrounding this position. The GT splice donor site closest to 19,150,856 is located at 19,150,858 to 19,150,859, which is supported by multiple lines of evidence such as our NCBI-RefSeq genes, our spawn alignment of D-melanogaster proteins, and the Gemoma, NSCAN, and Augustus gene predictions, as well as our RNA-seq read coverage from samples of adult females and adult males. Since we have located the exact coordinates of our splice donor for CDS1, this also means that our ending coordinate for the coding region of CDS1 will be right before our splice donor at position 19,150,857. Now, let's zoom out far enough to see the entire length of CDS1 and confirm which reading frame we are in. We can see here that reading frame 1 has stop codons in it. However, reading frame +3 has an open reading frame, meaning that there are no stop codons, and it also has the start codon we need at the beginning of our CDS. Using T-BLAST-N in Part 5, we determined the approximate location of CDS1 was 19,150,809 to 19,150,856. Using the Genome Browser in Part 6.1, we verified the start codon of CDS1 was in frame +3 at 19,150,809 to 19,150,811. We visually inspected the region surrounding the approximate end of CDS1 and placed the splice donor site at 19,150,858 to 19,150,859, after which we confirmed frame +3 of CDS1 has an open reading frame. Now we need to determine the phase of the splice donor site for CDS1. In order to do this, let's enter NC underscore 052530 colon 19,150,849 19,150,859 into the chromosome range or search terms text box and click on Go. Here we see the last complete codon containing three nucleotides of CDS1 before the splice donor site codes for the amino acid tryptophan in frame +1, arginine in frame +2, and valine in frame +3. The splice donor site at 19,150,858 to 19,150,859 is in phase 0 relative to frame 1 because CDS1 ends in a complete codon and thus has no extra nucleotides. In contrast, frame +2 and frame +3 don't end in complete codons, so they each have extra nucleotides. The splice donor site is in phase 2 relative to frame 2 because there are two extra nucleotides, which are GG, after the last complete codon and before our GT donor site. The splice donor site is in phase 1 relative to frame 3 since there is one extra nucleotide, a G, after the last complete codon and before that GT splice donor. We previously determined CDS1 is translated in frame +3, therefore, the last complete codon of CDS1, GTG, which codes for valine, is located at 19,150,854 to 19,150,856, and there is one extra nucleotide, a G, at 19,150,857 between the last complete codon and the splice donor site. Hence, the CDS1 splice donor site is in phase 1. An easy way to remember the phases is to think of the number of nucleotides being cut off by your splice donor or acceptor and is left inside our coding region. For T-blast N alignment in part 5 placed CDS2 at approximately 19,150,987 to 19,151,055. Therefore, we expect to find the splice acceptor site for CDS2 around position 19,150,987. To examine the genomic region surrounding the splice acceptor site of CDS2, enter NC underscore 052530 colon 19,150,987 into the chromosome range or search terms text box and click on Go. Zoom out 3 times and another 10 times to examine the 30 base pairs surrounding this position. There is only one potential canonical splice acceptor site, which is AG, in that 30 base pair region surrounding the start of the T-blast N alignment to CDS2. The splice acceptor site is located at 19,150,983 to 19,150,984 and is supported by the NCBI RefSeq genes and spawn alignment of demyelinating ester proteins, the Gemoma, NSCAN, and Augustus gene predictions, and the RNA-seq read coverage. Thus, CDS2 starts at 19,150,985, right after our splice acceptor. Now we need to determine the frame in which CDS2 is translated. To do that, enter NC underscore 052530 colon 19,150,980 dash 19,150,990 into the search terms text box and click on Go to zoom in on the region surrounding the splice acceptor site. Each frame can have extra nucleotides, 0, 1, or 2, between the beginning of CDS2 at 19,150,985 and the first complete codon of each frame. In frame 1, we have 2 extra nucleotides, GC, before the first complete codon, K, coding for lysine. Our splice acceptor site is in phase 2 relative to frame 1. Frame 2 begins with a complete codon, A, alanine, so it has 0 extra nucleotides, which means our splice acceptor site is in phase 0 relative to frame 2. Frame 3 has 1 extra nucleotide, G, before the first complete codon, Q, which is glutamine. So our splice acceptor site is in phase 1 relative to frame 3 because we have just that one extra nucleotide. Since we determined the CDS1 splice donor site at 19,150,858 to 19,150,859 is in phase 1 relative to frame 3, the CDS2 splice acceptor site must be in phase 2 to maintain that open reading frame after intron 1 has been removed. Since CDS1 had one extra nucleotide, that G, between the last complete codon and that spliced donor site, CDS2 must start with two extra nucleotides to join the extra one from CDS1 because we need three nucleotides to make a complete codon. Since CDS2 is in frame 1 and the first complete codon, AAA, coding for K, is located at 19,150,987 to 19,150,989, there are two nucleotides, GC, at 19,150,985 to 19,150,986, between the potential splice acceptor site and the first complete codon. Hence, we can determine that the CDS2 splice acceptor site is in phase 2. Extra nucleotides near the splice sites, G and GC, will form an additional amino acid, glycine, after splicing. Collectively, our analysis suggests that CDS1 ends at 19,150,857 with a phase 1 splice donor site, and CDS2 begins at 19,150,984 with a phase 2 splice acceptor site. The same annotation strategy can be used to determine the phases for the remaining splice donor and splice acceptor sites between CDS2 and CDS3, CDS3 and CDS4, and CDS4 and CDS5. Now we have finished reviewing the phases of splice donor and acceptor sites. Recall that to maintain the open reading frame across adjacent coding sequences, the phases of the donor and acceptor sites of adjacent coding sequences must be compatible with each other, meaning that the sum of the donor and acceptor phases of adjacent coding sequences must either be 0 or 3. Looking at the splice sites between CDS1 and CDS2, we see the splice donor phase of CDS1 is 1 and the splice acceptor phase of CDS2 is 2. Thus, the sum of the donor and acceptor phase for intron 1 is 3, and we can apply that same concept for the rest of our coding sequences.